All right, we are going to continue our lecture on cell membranes and transport. The accompanying slides for this video is also in that Google Slide folder that is linked on Moodle. All right, so let's cover some of these concepts that we didn't get to in class yet, but they will still be a part of Exam 1. We will do a review the next time we meet. Right, we quickly talked about the types of transport. Let me move this up here. There we go. All right, we talked about the types of transport, right? And we talked through simple diffusion, right? Simple diffusion meaning substances are moved across a concentration gradient. They don't need a protein to help them out with this. And then we touched upon facilitated diffusion, another type of passive diffusion. All right, excuse me, passive transport, passive diffusion, right? That is substances moving across a concentration gradient, but this time they need the help of a transmembrane protein, right? They can't move across the gradient themselves. Either they're too big, they have a charge, right? So they can't get through that hydrophobic interior. They need a protein to help them. We're also going to talk about active transport. Right? So active transport actually requires energy, and requires energy in that it's going against the gradient. Molecules don't like to go against the gradient, and so when they need to go against the gradient, they're going to require energy to do so. All right, so facilitated diffusion. Right? So again, I talked about facilitated diffusion needing a protein in order to get these molecules across the membrane. Right? So these transport proteins are transmembrane proteins, and they provide a passageway for larger molecules, molecules like ions or other hydrophilic molecules to get across the membrane. Now you can see in this image down here, I have two different types of proteins that we will talk about. One is a channel protein and one is a carrier protein, right? So if we're going back to that fence analogy, facilitated diffusion is like we have a fence, but there is a door in the fence that allows movement of different particles or different people to cross over that fence. And just like there can be different types of fences, different types of fence gates, there can be different types of transmembrane proteins. In this case, being our channel proteins and our transporters. All right, let's look at them a little bit closer. All right, our channel proteins are basically a transmembrane protein that is almost always open or available. It's an open passageway. Right, we can see on this image here that the molecules are simply using it as a gate, using it as a passageway to get across that phospholipid bilayer. Right, and again, we're following the concentration gradient. We are going from areas of high concentration to areas of low concentration. Now, some of these are just open passageways like we see in this image up above, but others actually are gated, meaning that they control when the gate is open or closed, right? So when the gate is closed, it prohibits the passageway of these molecules across the membrane, all right? So maybe the interior of the cell doesn't need any more of that type of molecule. It can close the gate and prohibit passageway. When the gate is open then, right, it stays open and acts just like this channel protein up above, right? The molecules will go through, again, areas of high concentration to areas of low concentration, right? So channels, they are passageways. The other type of protein that is associated with facilitated diffusion are transporters, right? So again, they're a transmembrane protein. They span the entire phospholipid bilayer. Sometimes they're also called carriers because they carry the molecule across, right? So unlike channels, when it's open, and it's always open, right, unless the gate is closed, but once the gate is open, it stays in that position and allows molecules to flow through, right? Transporters actually change their shape when molecules move through them. They go through something called a conformational change. So if we look at this image here, right, we have this transmembrane protein. It has this pocket to it, a hydrophilic pocket, right? So water-loving pocket. So molecules that are water-loving, like ions, other larger molecules, maybe like sugars, amino acids, nucleotides, they can go into this pocket, right? Once that pocket is filled, right? So let's say this is the pocket. Once the pocket is filled with our substance, all right, let's use a pen here, right? It's going to change its shape and allow the molecule to fall across or to move across. All right, I do have a quick, just kidding. My video's on the next slide. All right, but the main thing here is that unlike a channel, when it's open, it's always open in this position. Transporters will change their shape as things move across. All right, here is my video that I had just to be able to kind of watch this as it moves along, right? So I talked about it having a hydrophilic pocket, which we saw, that's where the molecule comes in, right? And then it's able to switch back and forth between these two conformations. And so in this image here, I think it gives you a good visual of how this happens. The solute goes into that protein changes shape, and that solute moves across the membrane, right? And it does this over and over again, right? But again, we're still talking about facilitated diffusion. So that means we're still moving these solutes across a gradient, going with the gradient from areas of high concentration to an area of low concentration, right? Because we're moving with the gradient, we do not need any energy. Just like simple diffusion doesn't need any energy, facilitated diffusion doesn't need any energy. All right, so we have these transmembrane proteins, these carrier proteins that change shape. We can have different types of them. All right, so we can have transporter proteins, like we just talked about. A molecule comes in, it changes its shape, the molecule comes out. That would be called a uniporter, right? Because it's only carrying a single molecule or a single ion at a time. We can also have a transporter that is a symporter, right? A symporter means that instead of just one molecule or ion coming into the transporter, there's two coming in at the same time. And once the two bind, we change our conformation and they move to the other side. All right, again, I kind of have a little video that demonstrates it here. So instead of just one molecule coming in, right, now we're going to have two. We see this small green one has bound there. It's waiting for the right other molecule. There it goes. Changes its shape. They're both released on the other side. So symporter, two or more ions or molecules transported in the same direction at the same time. We also have an antiporter. So an antiporter is where we're moving two or more ions or molecules, right? But they're being transported in opposite directions. So if we look at this image down here, right, we have this blue molecule coming in to the hydrophilic pocket of the membrane. Oh, pressed a button there, right? This protein, once it's bound to its first molecule, will change shape, and that blue molecule will cross the membrane, right? But now this transmembrane protein is going to stay open to that side until a different molecule binds to the hydrophilic pocket, in this case a red like rectangle molecule here. Then it will change shape and release the rectangle in the opposite direction. Right? So antiporter, two or more molecules, but they're going in different directions. Now again, even though they're going in different directions, they're still moving with that solute's concentration gradient. They are still moving from areas of high to areas of low, right? It just so happens that in the antiporter, the area of high to low for the blue molecule is different than the area of high to low for the red molecule. All right, so we've talked about passive transport, which includes simple diffusion and facilitated diffusion. They require no energy. Now, what if we need to move a solute across the concentration gradient, going against the concentration gradient? So now we're going from a region of low to a region of high. So if we're going from a region to low, going to a region of high, we're going against the gradient, because remember, solutes always like to go from areas of high to low, right? Now we're going from low to high. This is hard to do. It's energetically unfavorable. Molecules can't just do this on their own. They need an input of energy in order to allow this to happen. And this is what makes it an active transport, right? Active transport, we are going against the gradient, and we need a source of energy. All right, so if we're going back to our fence analogy here, we have our fence, right? There's no gate in the fence, right? But there is a way across. You just have to have somebody to help you out, somebody else to give you a push, give you that burst of energy to get up and over the membrane. All right, up and over the fence. All right, so this would be our active transport. Now, there are two different types of active transport. There's primary active transport, and there is secondary active transport. Both require energy, but in different ways. All right, let's look at active transport, primary active transport first. So primary active transport uses a pump, right? Uses a transmembrane protein that's kind of like a pump, and it requires energy. All right, so here we have our transmembrane protein. We're trying to move these hydrogen ions across the membrane. We're going from an area of low concentration to high concentration. We're going against the gradient, right? So these hydrogen ions will bind to this transporter protein, right? And the transporter protein isn't going to change its shape until we get some energy. Now we're going to use energy from our molecule ATP, right? This is an energy-giving molecule. We'll talk more about this molecule in a little bit. All right, so once this molecule, ATP, gives energy to our transport protein, now it will change its shape and release this hydrogen ion against the gradient to the other side of the membrane. All right, so that's primary active transport. Relatively straightforward. Secondary active transport, right, uses a separate gradient to drive movement, right? Uses a pre-existing gradient that had to have been set up using energy. All right, so if we look at secondary active transport, you might notice that this molecule that's going through a conformational change, I don't see any ATP around it, right? So it's not directly using ATP, but it's using the gradient set up by the initial primary active transport, right? Setting up this gradient of hydrogen ions using energy. All right, so we have this gradient of hydrogen ions. We set up this gradient by active transport, so energy had to be used. Now that this gradient is established, we can move on to secondary active transport. So we're looking at the image here. We have our hydrogen ions, these triangles, right? These hydrogen ions are going to bind to our transmembrane protein. And notice this is a symporter, right? Because we're moving two molecules in the same direction. Once the hydrogen binds to this transporter, then our other molecule, in this case, we're going to say sucrose, these round circles, are going to bind, right? Now, because this hydrogen ion is moving with its gradient now, right? Initially, it moved against its gradient in the primary active transport step of this, but now it's moving with the gradient. So because it's moving with the gradient, it will cause this protein to change its shape, right? Now, because there's also sucrose bonded, and sucrose is going against the gradient, when it changes its shape, right, sucrose will be let out against its gradient using this secondary active transport process. All right, so primary active transport uses ATP directly. Secondary active transport uses ATP secondarily, right? It uses a concentration set up by primary active transport. All right, recap here. All right, we have passive transport. Passive transport means no energy is needed. That includes diffusion, simple diffusion, and it includes facilitated diffusion. We're always going from an area of high concentration to an area of low. Now that also includes osmosis. We'll talk about that in just a second here. Now active transport requires energy because we are going against that gradient. We are going from an area of low concentration to an area of high concentration. All right, that was our recap of types of transport. I mentioned osmosis. Now let's take a closer look at osmosis. When I did put in this recap slide that might help you with your studying, right? What determines how a molecule can move across the plasma membrane? I've included this slide on the lecture from when we first started talking about transport in class. I'm including it again here because I think it's a really good review slide. All right, osmosis. Let's talk about osmosis. So osmosis is a type of simple diffusion, right? It doesn't require energy and it doesn't need any transmembrane protein. What happens with osmosis is that instead of the solutes moving, the water is going to move, right? So say we have a membrane, and this membrane is selectively permeable, right? It can allow water to move, but it can't allow a solute to move, right? It just doesn't have the means to allow the solute to move across the membrane. So what water is going to do is move across that membrane instead. And water is going to move according to the solute concentration, right? So the water follows the solutes. The solutes can't move, but the water can. If we look at our image here, right, we see on one side of our U-tube here, yes, that is what it's called, a U-tube, right, has many more molecules than the other side. Right? Now remember that the molecules, the solutes, cannot move, but the water can, right? So the water is going to move to the other side in order to get an equal concentration on either side here. Right, let me tap that into place here. Oh, I guess this is as small as it gets. All right. So again, we're kind of thinking about this as like a ratio of water molecules to solute molecules, right? When we have fewer solutes to water molecules, we're not in equilibrium, right? One side has a different ratio of solutes to water than the other side. So water wants to move to equalize that ratio on both sides. Right? And we see that down here represented in more of a molecular form. We can see the molecules of water. We can see the molecules of the solute that cannot move across the membrane. And we see on this side there are far more water molecules to solute, right? So osmosis is going to cause our water molecules to move across the membrane to the side that has more solutes, until this ratio of water molecules to solutes is roughly about the same on either side, right? Now that might mean if we look at this U-tube here, that one side might have more water than the other side. But that's okay because the ratio of water to solutes is the same, even though the amount of water is not. All right, so there are some important terms to know when we talk about solute concentrations associated with the cell. So these terms only apply when we are comparing two solutions together, right? We can't just say a solution is hypertonic, which means the solute concentration is higher than a water concentration. If we said the solution was hypertonic, you wouldn't know much, right? Hypertonic compared to what? You need to compare it to another solution. So let's go over these terms. Our first term is isotonic, meaning the solute concentration across a membrane on both sides is equal, right? So the solute concentration is equal. We can see that in this top image here. Hypertonic, right, means the solute concentration is higher than the other side of the membrane. All right, so if we look at the second image here, right, we see that on the outside of this cell, right, it is hypertonic compared to the inside of this cell. It is hypertonic because there are more solutes. Now on the opposite end of this, we have hypotonic. This means the solute concentration is lower on one side of the membrane than the other, right? So again, if we're looking at the outside of our membrane here, there are fewer solutes, the concentration is lower. Thus, the outside of our cell is hypotonic compared to the inside, right? Now, if I still look at this bottom image, right, I could look at it in a different way, right? I could say that the inside of the cell is hypertonic to the outside of the cell because the inside of the cell has a higher solute concentration. All right, let's take a look at some real life examples of why this matters. Say we have a red blood cell. All right, we put a red blood cell in a hypertonic solution, right? What that means is the cells are going to start to shrink because the water is exiting the cell, all right? We placed it in a solution that has more solutes outside of the cell than inside of the cell. Now, because of osmosis, this means the water is going to move towards the solutes, towards the side of higher concentration of solutes. It will move outside of the cell, right? Thereby shrinking that cell down, right? Very bad for our cells. We don't want that to happen. On the opposite end of the spectrum, we're going to place a healthy red blood cell in a hypotonic solution, meaning the solution around the cells has fewer solutes than the inside of the cell, right? When this happens, again, water follows the solutes. Water is going to go inside of our red blood cells because that's where the higher concentration is. And what can occur is that there is just so much water now inside of our red blood cells that our red blood cells can swell and even burst, right? Again, we don't want that to happen. This is why our bodies tend to maintain homeostasis, tend to regulate the amount of solutes in our bodies between different membranes, between different areas. Same thing can happen to a plant cell, right? We place it in a hypertonic solution. The water is going to want to leave the plant cell. But remember, the plant cell has this cell wall, which helps it maintain its shape, right? So the cell itself is not going to shrink, but the plasma membrane will start to cave in or shrink away from the sides of the cell wall. All right, so we're kind of losing a little bit of rigidity of that plasma membrane against the cell wall. It's no longer pressing up against the cell wall. It's starting to shrink away. Opposite happens when water enters the cell. Again, notice the overall shape doesn't change too much, but now we can see the plasma membrane is really pushing up against the side of that cell wall. All right, one last thing I wanted to mention about water movement. We talked about osmosis, right? And osmosis is a type of simple diffusion, right? It does not need a protein. Sometimes there are cases where a cell just needs to bring in a lot of water, right? Think about root cells of a plant. One of their main jobs is to take in water from the soil. Now, it could do this via osmosis, right? It could do this with simple diffusion, but that process can be slow when you need to take in really large amounts at a time. So there is a very specific transmembrane protein, and we see that at the top here, called an aquaporin. This aquaporin, right, is a channel protein, right, that allows much larger quantities of water to move across the membrane at a time, right? It speeds up this process. We greatly increase the rate at which water can move across the membrane. And again, they're found in these specialized cells that really need to bring a lot of water across their membranes, like plant root cells or in our kidneys, right? We need to move a lot of water across our membranes of the cells of our kidneys. We might find these aquaporins, right? So when water uses an aquaporin, it's no longer simple diffusion, but now we're talking about facilitated diffusion because a protein is involved. All right, that was my last slide. We will review this in class with some review questions. Make sure you understand this material and ask me questions if you do not, because all of this material will be fair game for exam 1.